The next type of plasmogamy in Phycomycetes is by gametangial copulation. Gametangial copulation is found in terrestrial mucorales. In the gametangial copulation, the uniting gametangia may be morphologically similar or dissimilar. If they are morphologically similar, then it is isogamous gametangial copulation, and if they are morphologically dissimilar, then this type of gametangial copulation is called as anisogamous gametangial copulation. In the gametangial copulation, the uniting gametangia lose their identity. In isogametangial copulation, as it occurs in rhizopus and mucor, what happens? The cases may be homothallic or heterothallic. Isogametangial copulation, it occurs in rhizopus and mucor. In these genera, both the cases may occur. There are homothallic species as well as in heterothallic species. In case of heterothallic species, the hypha of opposite strain come to very close to each other, and at the point of contact they form a short lateral outgrowth. This short lateral outgrowth is called as progametangium. Later on, the progametangium is divided into two parts by the formation of a septa, the anterior portion of which is called as gametangium, that is swollen portion, and the posterior portion is called as suspensor in both the cases of the gametangial branch. Then the intervening wall of the gametangia dissolve, as a result of which the protoplasmic content of both the plus and minus strain come to a fusion cell. In this fusion cell, the nuclei fuse to give rise to the zygospore. Initially, the zygospore is remains surrounded by a single wall. Later on, it becomes surrounded by two-layered structure. Outer structure is sculptured and dark in color. When the mature zygospore is ready to liberate, then the wall of the gametangia rupture, as a result of which this zygospore comes outside. So this is the case of isogametangial copulation in heterothallic species of rhizopus and mucor. Now another type of gametangial copulation is anisogametangial copulation that occurs, for example, in Zygorhynchus. In anisogametangial copulation, what happens? There is anisogamy in the sense that the fusing gametangia are of unequal size. The male gametangia is smaller and the female gametangia is larger in size. Their size also range as in the sense of suspensor also. After fusion, they form the zygospore. Zygospore is a thick walled structure. Third one is the anisogametangial contact. This is another type of plasmogamy, as it occurs in endogone lactiflua. In this case, the fusing gametangia never lose its identity. The male and female gametangia differ in the sense of their size. That is, their hypha differ in their diameter. In a similar way as in the case of gametangial copulation, the male and female gametangia are formed. At the point of contact, they form a pore, and through this pore, the male gamete gets entry into the female gametangia. Only other nuclei become degenerated, and there the fusion of only two nuclei. One is male nucleus and another one is female nucleus. Both these nuclei fuse to give rise to a structure that is called as zygospore. This develops in a structure that is formed by the female gametangia. There is a formation of a tube-like structure by the female gametangia, which enlarges to give the structure that is formed inside the structure in which the zygospore is formed. This zygospore remains surrounded by a sheath. So this is the case in endogone lactiflua, where the entirety of male gametangia and female gametangia is never lost. After this, such type of plasmogamy in Phycomycetes, karyogamy is immediately followed. As in the case of higher fungi, the karyogamy is delayed and there is a long dikaryotic phase. In case of lower fungi, there is no such dikaryotic phase, as the plasmogamy is immediately followed with karyogamy. Now, after karyogamy, there is formation of zygospore. On getting the favorable condition, the zygospore shows germination. The germination of zygospore amongst the Phycomycetes fungi is of three different types. One is the case in which the zygospore germinates after meiosis. When the meiosis occurs at the time of germination of zygospore, a haplomycelium is produced. Haplomycelium means the mycelium having haploid nuclei. Such type of mycelium, that is hypomycelium, is produced in case of monoblepharis. This is the case of haplomycelium, when the zygospore germinates by meiotic division. In the second type, if the zygospore germinates without any meiosis, then the mycelium contains the diploid nuclei, and in such cases a diplomycelium is produced. Diplomycelium is produced in the case of allomyces, for example. Second type of germination of zygospore is when the zygospore undergo meiosis to form the meiospores, as it happens in albugo and pythium. In albugo, when the zygospore germinates, it forms a sessile vesicle. Inside this sessile vesicle, the meiospores are formed by the process of cleavage. In pythium, when the zygospore germinates, it forms an exit tube. On the exit tube, the vesicle is formed. In this vesicle, the meiospores are differentiated. In this way, albugo have a vesicle that is sessile, while in the case of pythium, the vesicle is stalked. This is second type of germination of zygospore among Phycomycetes. Now the third type of zygospore germination is by producing promycelium, as in the case of Plasmopara and Mucor. When the zygospore germinates, this is the example of Mucor or Rhizopus. When it germinates, it forms a promycelium. In this promycelium, at the tip of this promycelium, the sporangia is differentiated. Inside this sporangia, the spores are formed by the process of cleavage. The spores may be motile or non-motile. Motile spores are produced in Plasmopara and spores are formed in Mucor. So these are the three types by which the germination of zygospore among Phycomycetes occur.